one of the major things that meeting all these microorganisms does. It gives you a huge repertoire. The bigger, the more diverse the organisms you have met, the better, and the broader your repertoire. Now, if your repertoire is big enough, because all life forms are ultimately built from the same sort of building blocks, most of our genes, for instance, evolved in microorganisms. It may seem extraordinary, but the first versions of those genes, more than half of our genes, evolved in microorganisms. They've become somewhat modified, as we've adapted them to our purposes. But it means that all life forms are ultimately built from similar building blocks. We know that many of the organisms that we need to be exposed to really come from two sources. They come from mother and other family members, and they come from the natural environment. But there is a clinical trial, well, I suspect several clinical trials running, of doing fecal microbiota transplants into autistic children. And there are rumors that at least one of these trials may be looking encouraging. Although ultimately, initially, the disease was caused by a defect in the immune system, it is now, the symptoms are actually caused by the change in the microbiota. And of course, this raises all sorts of questions about situations in human, because all sorts of things can temporarily upset your immune system. But then you may have an abnormal microbiota, and this is that which is perpetuating the subsequent disease.
Hello, everybody, and welcome back to Chasing Consciousness. So today, we're going to be getting our heads around the idea that while hygiene has revolutionized modern health, too much hygiene actually weakens our immune system's development. Now, to understand why, we need to understand how the complex community of microbes in our intestines, on our skin, and in nature around us has co-evolved for thousands of years in symbiosis with our immune system. And we'll be mapping how, as our hygiene practices and food processing has increased, so the diversity of microbiota has dropped, eventually leading to the explosion of inflammatory disorders, mental health issues, and autoimmune dysfunction that we're now seeing in Western communities. But how to solve this problem without losing the great advances in healthcare achieved by the application of hygiene theory? Now, who better to answer this question and explain the subtleties than Professor of Medical Microbiology at University College London, Dr. Graham Rook. Now, Rook is a specialist in infection and immunity and has spent his entire career unfolding how the young immune system is programmed by the microbial background, identifying all the medical conditions that have been worsened by this failing immunoregulation associated with our impoverished biomes, and developing ways that we can update our lifestyle to restore the diversity required to sustain that healthy immune response. Now, in his new book, Evolution, Biodiversity, and a Reassessment of the Hygiene Hypothesis, he explains all of this and brings back his 2003 Old Friends hypothesis, which seeks to correct the overstretching of hygiene hypothesis. Now, we have got a show in the second series with phytotherapist Alex Laird about the dietary aspect of the microbiome, and that's called Mood Food, should anyone want to get really deeply into the food and diet side of all of this. Now, don't forget, listeners, that all of the books, the quotes, the scientific papers, concepts, and conversation points are exhaustively included in the show notes, should anyone want to check the authority of all of the research and the ideas that we're citing here on the show. On the website, there are even more, should I be unable to fit them all in, so please do use them. I'm a stickler for well-referenced claims, even if I don't agree with them all. And a quick thank you for all of your support on the project so far. Keep spreading the word by sharing the shows with your friends. And if you can give me a 5-star rating on iTunes and subscribe with the bell on YouTube, that also helps get the show in front of like minds. Also follow me on Facebook and Instagram to be kept informed about the new shows as they come out, and to join me and the other listeners in the ongoing conversation around all of these fascinating new emerging ideas in science. Now, I have been wanting to speak to Dr. Graham Rook ever since he was recommended by Alex Laird in the first microbiome episode called Mood Food. So I really can't wait to get into this. So let's go. Dr. Graham Rook, welcome to Chasing Consciousness. Thank you so much for coming on the show. How are you today?
I am very well, thank you, and very flattered that you want to interview me about this.
Well, as one of the world's specialists, it seemed a logical place to go. So, Graham, I love to start by asking my guests about the first big questions that rang in their minds as they first became conscious about the world in early adolescence. Can you remember any of your reflections from that time that might have some way influenced your future research?
Well, I certainly was always playing with microscopes. Right from early childhood, I had a battered old brass microscope, and I spent much of my time looking at pond creatures. So I think I got very interested in this smaller communities of tiny creatures living on our planet. But I didn't really get interested in the topic you're interviewing me about today until really much later in life, when I had been doing research on tuberculosis. And we got interested in organisms related to the tuberculosis organism, but harmless, that were found in soil. And we discovered that people were actually responding immunologically. They had immune responses to these microorganisms in the soil in the areas where they lived. You could almost tell where somebody was born and where they lived on the basis of which microorganisms from soil their immune systems recognized. And that was sort of an eye-opener to me. And then a colleague of mine, John Stanford, isolated one of these organisms, actually from mud in Uganda. And I started playing with it, and it became a kind of model system that we played with, and in particular with a neuroscientist in America called Chris Lowry and another neuroscientist in Germany called Stefan Reber. And we found that if you immunize animals with this organism, it would actually stop them from developing allergic disorders. And in this experimental model, if you stressed the animals, they developed a nasty colitis and they developed behavioral changes. But again, exposing them to this harmless mud organism blocked all of these changes. It stopped them from having behavioral changes as a result of the stress, and it stopped them from developing the colitis, which really covers much of ultimately what we will be talking about today. So
absolutely,
I was so struck by this.
This codependence is very much where we're going to be heading when we look into the microbiome itself. So before we get on to the implications of your research about how we should practice hygiene, and to your new book, Evolution, Biodiversity, and a Reassessment of the Hygiene Hypothesis, and before we get into the effect the microbiome has on depression and our state of mind, let's get into the gut itself, just to set the scene for the listeners. Now, when we speak about individual organisms, we might get confused into thinking that we're just individuals and that we're only human, rather than thinking of ourselves as a hugely diverse ecosystem in ourselves that provides a home for all these kinds of species of bacteria and viruses and fungi. Now, Graham, can you just start by describing how this finely balanced symbiotic relationship evolved? And perhaps more importantly, how it programs, as you put it, our immune systems.
Yes, well, as you say, I mean, the fact is that more than half of all the cells in our body are in fact bacterial cells, not human cells at all. And because these bacteria consist of, and other microbes consist of multiple species, it means that in terms of the genes and metabolic pathways encoded in those genes, there's far more of microbial origin than of human origin in a human individual. So how did this happen? Well, you really have to look back in evolution. Cellular life, by which in this context I mean bacteria and things called archaea, which look rather like bacteria, evolved about 3.8 billion years ago. And then a couple of these organisms merged, one got inside the other, about 1.5 billion years ago, and became what we call the first of the eukaryotic cells, the cells from which humans ultimately evolved. So we ultimately evolved from bacteria, but we also evolved in a world, a kind of soup of bacteria. Even now, the total weight of bacteria and other microorganisms on this planet is far greater than the total weight of humans. You might think humans are big, heavy things, but oh no, there are so many microorganisms that there must be at least 100 times greater weight of bacteria on the planet than there is of humans. Bacteria are second only to plants in terms of the total weight of material out there. So we evolved from bacteria, but we also evolved in a soup of bacteria. Now, if you go to, for instance, the insects, which evolved about 480 million years ago, you find bacteria in their gut. Well, that's inevitable because they're living in a soup of bacteria. But those bacteria are separated from the insect by a kind of chitin barrier. Chitin is that sort of hard material that you see on the outside of a prawn or an insect. But this was a chitin membrane, and the bacteria were in there, separated from the insect. But when you get to about 200 million years ago, where we get true vertebrates existing, obviously we're getting closer to us now, what you find is that the chitin barrier has gone, and the bacteria are now closely associated with the gut wall. And in fact, some of them are actually in the mucus on which the gut wall is covered. And what's more, they eat that mucus. We are feeding these organisms. They live on the mucus and they provide signals that affect our physiology. So it then emerges that these organisms that we are now closely associated with our physiology have actually co-evolved with us. And recent studies of humans spreading out, the humans leaving Africa and spreading out across the planet, you find some of the strains of bacteria in our guts have obviously gone with them. And what is more, they're losing some of their genes, so that some of the bacteria that live in our guts now have lost most of their genetic material, because these were genes that they needed to live out in the mud, but they don't need them now, they're living in us. So they're becoming obligate symbionts, to use that sort of technical term, that they need us. But similarly, we need them. And in fact, what is so fascinating is that it's rather the same solution that plants have come up with. Because if you think about it, a plant lives in soil. Plants put polysaccharides and mucus-like things onto the surface of their roots. Bacteria are attracted by that. They eat this polysaccharide material, so they're fed by the plant, but at the same time, they're giving various signals to the plant and various nutrients to the plant. So it's beneficial to the plant and to the bacterium. We have exactly the same situation, except that rather than sitting in the soil, we carry it around with us in our guts. But it's an interesting parallel.
Mm.
So,
and how does that then lead to the creation of the immune system? So the development of the immune system in this evolutionary story. You use the word programming. Tell us about how that evolved.
Well, yes, you see, the immune system that we now have, which really didn't evolve until this phase of development of the vertebrates and mammals, is what's called the adaptive immune system. Now, the point about this is that if you go back further in evolution, obviously there was some kind of an immune system, but what you inherited was molecules encoded in the DNA, which would recognize common bacterial components. But that's all very well if the bacteria keep evolving, and what is more, if you've got huge numbers of them in your gut in close association with the gut mucosa, which is the situation we have now, because the bacteria can evolve very quickly, whereas we can't evolve at the same speed. So we needed some way of keeping up with the rate at which bacteria and other microorganisms can evolve. So we developed what is called the adaptive immune system. Now, the adaptive immune system is mind-boggling. It's absolutely incredible. What actually happens is that lymphocytes, some of which will make antibodies and some of which don't make antibodies but become T cells, but which also can recognize foreign things, these lymphocytes undergo mutation, deliberate mutation. So in each newborn baby, you've got this massive degree of mutation of these lymphocytes to produce a new repertoire, because each lymphocyte makes a different antibody or has a different receptor for bacterial components. So you develop this new repertoire, which then has to be censored. You have to choose from that repertoire those cells which actually recognize something present in your environment. There's no point in filling it. We'd be walking bags of lymphocytes otherwise, if we kept them all. We just want to keep the ones that are useful to us. So it's incredibly clever if you think about it, because it means that each generation generates a repertoire that is appropriate to the environment into which it has been born. Now, what happens is that if the lymphocyte doesn't recognize anything in your environment, it is eliminated. But if it recognizes things in your gut microbiota or other microorganisms that you eat, then it is retained and becomes part of your repertoire for recognition of other things or infections or whatever in the future.
How long does that tuning take at the beginning of human life?
Well, the most important phase is infancy. That's why the most critical period for getting the right microorganisms on board is the early months and years of life. But that's only one of the major things that meeting all these microorganisms does. It gives you a huge repertoire. The more diverse the organisms you have met, the better, and the broader your repertoire. Now, if your repertoire is big enough, because all life forms are ultimately built from the same sort of building blocks, most of our genes, for instance, evolved in microorganisms. It may seem extraordinary, but the first versions of those genes, more than half of our genes, evolved in microorganisms. They've become somewhat modified, as we've adapted them to our purposes. But it means that all life forms are ultimately built from similar building blocks. So if you've got a huge repertoire of lymphocytes that recognize something from the environment, some microorganisms from the environment, with a little bit of luck, some of those will recognize some infection you've never even seen before. For instance, the HIV virus. We know that is the case now, that if an animal or a human has encountered enough microorganisms of a harmless type, there may be within its repertoire cells that can recognize something unknown and dangerous like HIV. So developing the repertoire is incredibly important. But the other really important thing, which is really, I think, the central theme of this discussion, the other really important thing that meeting these microorganisms does is turn on the police force of the immune system, the regulatory arm of the immune system, because the immune system is incredibly dangerous. It is perfectly capable of attacking your own tissues, or it can do stupid things like attacking the neighbor's cat that you happen to have breathed in a bit of dandruff from, or grass pollen, or whatever. You don't want it doing that. You don't want it attacking your own tissues, which is what, of course, happens in type 1 diabetes, where the cells that make insulin are destroyed by the immune system, or multiple sclerosis, where your brain tissue is being attacked by your immune system. So it's absolutely crucial to turn on the regulatory arm of the immune system. There are specialized cells within the immune system whose job it is to stop inappropriate immune responses and to terminate inflammatory responses when they're no longer needed.
Just about what we're going to get into, isn't it, Graham? Because we're going to be talking a lot about that. Before we move on to that, I would like to just quickly talk. You mentioned the mucus there. Sorry if this is a bit squeamish for some listeners out there. I hadn't until recently understood how important that mucosal lining was to our immune system, because it's packed with this really crucial secretory IGA. Can you explain a little bit more why this mucosal lining is so important to our immune response?
Well, yes, because we've got this huge community of microorganisms in the gut, and you want them to stay there. I mean, there are plenty of organisms in the gut, which, while harmless when they're there, would kill you if they got into your cerebrospinal fluid, for instance. So, firstly, the immune system in the gut has got to stop organisms or excessive numbers of organisms from crossing the mucosa and getting into you. But it's also a question of farming of the microbiota. I mean, literally, that's the term I tend to use, because we've got this vast number of organisms there. We want the right ones, we don't want the wrong ones, and the immune system actually can eliminate organisms it doesn't want in the gut. And you mentioned immunoglobulin A, or IgA, which is one of the mechanisms the immune system uses in the gut to limit numbers of certain types of organism. There are many other immunological mechanisms happening in the gut as well. A whole range of peptides that are released by cells of the gut epithelium, which affect certain organisms' survival and so on. So we need to keep this community of bacteria under control. The immune system farms it. But we also need to maintain the sort of two-way communication, which I'm sure we're going to talk about, between those organisms in the gut and the brain and other organs. So it's a very delicate and clever process.
So I think we've set the scene pretty clearly about how evolution designed this. Now let's get into modern life. Now, our research shows unanimously that there is a massive difference in the diversity of species in our gut and on our skin compared to our hunter-gatherer ancestors and the few remaining hunter-gatherer communities that we can study today. Can you map out the main reasons why this impoverishment has occurred and the corresponding set of pathologies that are exponentially rising in Western communities as a result?
Well, yes, indeed. I mean, if you think about the hunter-gatherer, for instance, they had massive seasonal changes in what they were eating. So, there was the season when they did hunting and the season when they would have been mostly gathering. We don't do that anymore. Our diets are much less varied. Because of modern transport and so on, we can eat pretty much the same thing every day of the year. But there are a whole lot of other more specific mechanisms as well. There's the question of how one is born. We know that many of the organisms that we need to be exposed to really come from two sources. They come from mother and other family members, and they come from the natural environment. Now, let's start with mother. The organisms you get from mother, you first start picking them up during the process of birth, because many of those organisms are present in the birth canal. And it may not be an evolutionary accident that the anus is very close to the birth canal. So some gut organisms inevitably contaminate the baby on the way out. And we know now there are at least 20 studies showing that being born by cesarean section limits the rate or slows down the rate at which you develop your maternal type of gut microbiota. And then breastfeeding, amazingly, breast milk is not sterile. Breast milk actually contains certain microorganisms, and it also contains certain molecules which, interestingly, cannot be metabolized by the baby. That's not why they're there. They're there because they can be metabolized by organisms that it's a good thing for the baby to have in their gut. So in other words, you might be called a prebiotic is the sort of term for that. So mother's milk provides some microorganisms, and it provides encouragement for the right kinds of microorganisms. So all of these factors I've mentioned so far are to do with the mother. And obviously in some families, you may be born by cesarean section, and you may not have breast milk, in which case this is going to limit your exposure. But then there's also the question of exposure to the natural environment. Here, the really good studies have been farming studies, for instance, where people who are brought up on traditional farms have much better immunoregulation than people who are brought up, for instance, on highly industrialized farms, where the farmer never gets near anything except for an air-conditioned tractor with high fidelity sound, and it's not really contact with animals. There may not even be any animals, and then exposure to the environment is also a problem of socio-economic status. And I think this is something I'd like to emphasize as we go through, that many of these problems with the development of an appropriate microbiota are associated with low socio-economic status and poverty for some very good reasons. Firstly, you may not have so much exposure to green space. Wealthy people tend to have a house in the countryside, so they get exposure to green space, or if they live in the middle of London, they have a garden anyway. So again, exposure to green space. So these microorganisms coming from the environment, they are going to pick up. Whereas somebody living on the 24th floor of a tower block will have much less exposure to green space. They may also have a poor diet and less varied diet. And having a varied diet is important in maintaining a varied microbiota. And they may also smoke. Smoking is closely associated with low socio-economic status in this country, and I think increasingly in many others. And smoking has a terrible effect on the microbiota, not only in the airways, but also in the gut. And then there are also things like exposure to pollution, because we now know that air pollution, which has all sorts of detrimental effects on health, through many different mechanisms, evidently, but one of the mechanisms appears to be curious effects, similar to those of smoking, on the nature of the microbiota. So there are a whole host of factors that are limiting our exposure to microorganisms, but roughly one can classify them as less exposure to microbiota from mother and less exposure to microbiota from the natural environment. And then, of course, there's antibiotic use. For instance, we know that there's a linear relationship between the number of exposures to antibiotics in infancy and the likelihood of allergies or of obesity. And obviously, antibiotics play havoc with your microbiota.
You mentioned there immunoregulation. Just talk us through how that happens and give us a list of these things that are coming up, terribly often these days: autoimmune disease, allergies, chronic inflammation situations. Talk us through how immunoregulation works in a healthy person and why that's not working and leading to this terrible list of pathologies.
Yeah, I mean, there are specific pathways within the immune system that have the job, I mean, literally a sort of military police of stopping responses to self. For instance, components of your own tissues will tend to be recognized by cells that have this regulatory function and will switch off immune responses. Now, what is so interesting in relation to what I was saying about exposure to green space is that it turns out that many of the organisms that turn on the regulatory pathways of the immune system and expand what are called the regulatory T cells, or Treg, turn out to be organisms that you find in the natural environment, spore-forming organisms, for instance. And so there's a close link with this exposure to the natural environment and the development of good regulatory pathways. So I don't know, you wanting me to go into very detailed immunology about all of this?
Well, no, it was just to understand clearly this idea of immunoregulation, because it seems like the problem that we're having is an overreactive immunoregulation. So I wanted to understand what's going wrong there that creates the overreactivity. But maybe we can just get that out of the next question, because it seems to me that the inflammation is the kingpin here. And we're seeing an exponential explosion of these overreactive inflammatory diseases in the West. So can you speak about how it's making errors by not switching off the inflammatory response when it's required? Because your research shows that quite often the immune response, the inflammatory response, just stays switched on even after the danger has been removed. What's malfunctioning there?
Well, in fact, we're talking about two slightly different things here. There's the specific responses against things like allergens or your insulin-producing cells or your brain cells and so on. So there's that kind of specifically targeted inappropriate immune response, which should be turned off by these regulatory lymphocytes. And there are a whole host of mediators that are anti-inflammatory and which one can measure. And one finds, for instance, more of that being produced by the people who have been on the traditional farms rather than the ones on the super-industrialized farms, for instance.
Yeah.
But then you've mentioned the other problem, which is that in communities in developing countries, what you tend to find is that if something causes an inflammatory response, which you need, like an infection, I mean, you need an inflammatory response because that's part of the immune reaction to the infection. In some countries, you find that the markers of inflammation go up and then switch off again once the job is done. But what one finds is that in communities in this country and the United States, if you measure some of the markers of inflammation in the peripheral blood, you find that they're just not going away. there's a sort of constant level of background inflammation. And now exactly which immunological mechanisms are involved in and are failing in that situation, I'm not sure that I'm able to tell you. I don't think we fully understand it. But what we do know is that having a persistently raised markers of inflammation in your peripheral blood without any targeted organ, there's nothing to tell you there's anything wrong with this individual. They're not getting allergies, they're not getting an autoimmune disease, but there's this background level of inflammation detectable by measuring these markers in the peripheral blood. What we do know is that these people are at risk of, or at greater risk of, things like cardiovascular disease, depression. Those are the main ones I would mention in that context. So we need to understand a lot more about how to turn off that background inflammation.
Well, it's very sad that science hasn't understood that background inflammation yet. I was really, really hoping you had some solutions for us there, Graham. But I know that a lot of research is being done. Now, regarding the importance of the microbiota to our mental and physical health, some of the situations you've been mentioning as we're going along here, there are some particularly fascinating results from my studies that show how the introduction of microbiota from, say, an obese subject into those of another microbiome leads to the second subject becoming obese. And the same results applied for the introduction of microbiota from depressed or happy subjects into others, and those other subjects became depressed or happy. Now, can you describe these results and the massive implications that they have for how in future we might treat both mental conditions like depression and psychosis, but also metabolic conditions like obesity?
Yes, these are incredibly important observations because in a way they answer the question of whether changed microbiotas are the causes or the consequences of the diseases. Because if you stress somebody, their microbiota changes. So how does one sort this out? Well, these transfer experiments do the job beautifully. So what they do is they take mice that are germ-free, or rats. That's mice that have been born by cesarean section into a sterile environment. And so they don't have any bacteria in their guts. And then you transfer into these animals bacteria from the guts of humans who are perfectly normal or who are depressed, and look at the behavior of the animals. Now, you'd be surprised, but in fact, a mouse that receives microbiota from a depressed human behaves differently from a mouse that receives microbiota from a non-depressed human. And as you say, you can do the same thing. In fact, it's been done with schizophrenia as well. I don't know quite what a schizophrenic mouse looks like, but its behavior is different from that of the mouse that received the microbiota from a normal human. The obesity thing is very clear-cut. You put the recipients of microbiota from a fat human or a thin human on exactly the same diet, but the ones that had the microbiota from the obese human put on more weight than the ones that had the microbiota from the skinny human.
Is this leading to changes in treatment methodology?
Well, maybe eventually, but just let me say that it's also been done with allergy now, and there's a slightly more complicated protocol. But I think, in a way, the most important experiments of all are ones that we haven't mentioned yet. These are experiments where you simply alter a gene in the immune system of an animal. Basically, you're impairing and making the immune system abnormal. And then that animal gets sick. But then the astounding thing is that you take the microbiota from that animal and put it in a germ-free animal which has not been genetically modified. In other words, the recipient animal is a perfectly normal animal, but it gets the microbiota from the immunologically abnormal animal. And believe it or not, that recipient gets sick as well. So what it's telling you is that although ultimately, initially the disease was caused by a defect in the immune system, it is now the symptoms are actually caused by the change in the microbiota. And of course, this raises all sorts of questions about situations in human, because all sorts of things can temporarily upset your immune system. But then you may have an abnormal microbiota, and this is that which is perpetuating the subsequent disease. So in a way, those are the most important experiments of all, I think.
And the implications are just so massive for how we treat in the future. Tell us how the medical establishment is responding to these results. Are there attempts to introduce new methodologies into how we treat this stuff?
The problem is that it is enormously complicated. That really is the problem. It may not, you see, it's not perhaps so much the actual microorganisms that matter, the microbiota, it's more their pattern of metabolites. These are the small molecules that you are getting from your gut. So the total metabolome, as we call it, that you are exposed to is the molecules that your system, your cells are making, plus the molecules that these microorganisms are making. And that is the metabolome. Now, there may be more than one way of achieving the appropriate metabolome. I mean, exactly what is the right pattern of microorganisms for you is going to depend on a variety of things like your diet and on your genetic background. So it's not easy. There's no such thing as saying, oh, this is the right microbiota for all human beings to have. It just isn't so. It's going to be different for different people, and it will depend on what you're eating. Those hunter-gatherers going from the hunting season to the gathering season will have had completely different microbiotas in a different season because they were receiving different microorganisms. Mm. And then because there are so many species, there's a lot of things we don't know. For instance, there might be some microorganisms that make a specific molecule that we need to function normally. And to take a slightly absurd example, there's a funny little thing called the zebrafish, which gets studied a lot because it's very small and transparent, so you can see what's going on. And the zebrafish needs a particular microorganism because that microorganism makes a particular protein which acts as a signal for the proliferation of the cells that make insulin. So it doesn't make enough insulin if it doesn't have that particular microorganism. So it needs that signal. Now, amongst the thousands of different organisms in our gut, there might be one or two that we need because we have evolved to require a particular signal from that organism. But we haven't identified such organisms yet. So if we had, we could find out if that one was missing in people with a particular disorder and put it back. But then there's the reverse concept as well, which is that there might be certain organisms which produce molecules that are toxic to us. I mean, every now and then you see a paper saying, we found it, rheumatoid arthritis is occurring only in people with organism X or organism Y, or the same thing you might see for diabetes or whatever. Now, sooner or later, one of those studies will be correct. Maybe some of the existing ones are already correct. I'm not sure yet, because it's very, very difficult. But of course, if that's the case, then one would try to eliminate those organisms. So you see, amongst this forest of thousands of different organisms, there are some that maybe we need to be without, some that we need to have, but we simply don't know yet. So we know the more general thing, that we need some that produce what are called short-chain fatty acids, and we need some that produce certain metabolites of bile salts, which are also necessary for our physiology. We need some that produce appropriate metabolites of tryptophan. So we know broadly various categories of molecule that we need to be produced by our gut microbiota. But we don't know whether there are some individual species with really important functions.
Now, coming back to the list, do you think that these results that you're talking about regarding depression, regarding obesity, psychosis, do you think that they merit further research into other conditions we're wrestling with at the moment, like autism or Parkinson's or Alzheimer's, or is that just going too far?
No, I mean, absolutely. There needs to be more research in these areas. But one would have to stress, though, you see that people with any of these diseases are stressed, which changes the microbiota anyway. And autistic children tend to be very picky eaters. So they're eating a funny diet. So that's going to affect their microbiota. Moreover, in all of the disorders you've mentioned, I think, Parkinson's, Alzheimer's, and autism, there is evidence of inflammation in the brain. Now, we know that the gut microbiota plays a role in the regulation of inflammation. So at one level or another, they're almost bound to be involved. But I guess your question really is: could there be a closer link, more than just these general points? And the answer is,
and potential treatment as well.
Yeah, entirely possible. For instance, Parkinson's disease, there's often gut symptoms, and in fact, many people now think it begins in the gut. Mm. And one interesting point, of the relevance of which I really don't know yet, is that there's a gene that's involved in a rather nasty, very early onset form of Parkinsonism, which turns out that gene in fruit flies, okay, it's a long way back in evolution, but in fruit flies, that gene is involved in regulation of the microbiota. That's kind of fishy, isn't it? And then if you look at autism, they often have gastrointestinal symptoms. And there is some evidence that in some of these children, treatment with the antibiotic vancomycin has, at least for a while, a beneficial effect. There's evidence that if you do a fecal microbiota transplant from autistic children into experimental animals, this can cause changes in the regulation of certain genes in the brain, which are thought to be involved in autism in humans. So it's looking as though there's something going on there. But again, it's the sheer complexity of the thing, which means that there's no real handle on it yet. But there is a clinical trial, well, I suspect several clinical trials running, of doing fecal microbiota transplants into autistic children. And there are rumors that at least one of these trials may be looking encouraging. So I would be surprised if we don't find that these areas of research become very valuable in the future.
And the developer of the GAPS diet, I believe she developed it for her child, actually, who was autistic, and saw extraordinary results just from changes in diet there. So there's definitely lots and lots of space for research here, isn't there? Now, Graham, I want to move on to the sort of trickier part of this discussion that really requires a very nuanced and clear balance of the contributing factors not to be misunderstood. Now, your new book, Evolution, Biodiversity, and a Reassessment of the Hygiene Hypothesis, tackles the need for a reassessment of our approach to hygiene. Now, looking at your data, many people could think that we were living an over-sanitized life for the promotion of our gut diversity. And yet, antibiotics and hygiene have been massively responsible for the drop in deadly disease. In the book, how do you argue we should manage this fine reassessment, retuning, to allow the reprogramming of our overreactive immune systems?
Well, I think that the first thing I need to say is that there are actually two separate things going on here. And I think that there's been quite a lot of confusion.
Well, this is what I wanted to clarify, because I think it's very important the public understands which is the right way to go.
Yeah. Firstly, we've been talking mostly about the question of regulation of the immune system, the fact that all these chronic inflammatory disorders like inflammatory bowel disease and autoimmunity and allergies and persistent chronic inflammation, we've been talking about the regulation of those and what is going wrong, why these chronic inflammatory things are happening. So that's one thing. And we know that in order to get the regulation going correctly, we need exposure to mother and to the natural environment, where the organisms that turn on the regulatory pathways are mostly found. But of course, there's something else going on, which is preparing the immune system for fighting off infections. And that's rather a different issue. And it's worth remembering that back in the 1930s, it was noticed that if you have one infection, this may make you somewhat resistant to another infection. In fact, this was done with cows and guinea pigs. But nobody really believed that or nobody took it seriously, because once we started to think in terms of the adaptive immune system, which is very specific, you have a response to one particular organism, it's not relevant to the response to another organism. But then the fact that you do get some cross protection between one thing and another emerged again in the 1980s, when a wonderful Danish group made the weird observation that when they vaccinated people in Africa with a live measles vaccine, the increased survival of the children was much greater than could be accounted for by the incidence of measles. In other words, the measles vaccine was actually protecting them from something else. And it's then subsequently turned out that most, perhaps all, live vaccines have this non-specific effect. For instance, BCG vaccine, the vaccine against tuberculosis, also causes increased survival, far greater than its not very large effects against protecting you from tuberculosis. And just recently, there's been a clinical trial of BCG in elderly men, I think, which has shown that if you give people the BCG vaccine, they are less likely to get respiratory infections. In other words, less likely to get virus infections. So all of this goes back to the 1930s again, where they realized that, in fact, to some extent, having an infection does cause cross-protection against other infections. Now, this is involving mechanisms quite separate from the regulatory pathways we were talking about earlier. And in fact, the whole thing is now being worked out in some detail, and it's got renamed. I've forgotten what it has been renamed. Trained immunity. People are now calling it trained immunity. And what it is activation of sort of putting the immune system on alert. It's raising the background level of alertness of the immune system. In fact, for the more scientifically minded members of the audience, it's done, in fact, by epigenetic changes to the stem cells that give rise to macrophages and monocytes and so on. So you have a more active communities of cells floating around in the system. So to some extent, then, having infections protects you from infections. But of course, there's a problem here, because you've got to survive the infection. Clearly, it's better, therefore, to get this enhanced activation of the immune system without the risk of dropping dead in the process. And that's why I mentioned the vaccine discoveries, because what we now know is that this protective effect of getting infections against other infections can be mimicked by vaccines. In other words, you don't need to run the risk of dying from measles in order to get the survival benefit, the benefit of increased resistance against other infections. All you need to do is have the measles vaccine. And of course, what this kind of work is showing is that the anti-vaccine people are more desperately, horribly, sadly, tragically wrong than we already knew they were, because vaccines, or at least the live vaccines, are activating the immune system in ways that help protect you from other infections. So there are two different things going on here then. There's the turning on the immunoregulatory pathways, which seems to be organisms from mother and from natural environment, and there's turning up the background level of alertness of the immune system, which does have some input from getting infections, but which vaccines can replace. Now, of course, the fact that we're getting such huge numbers of infections overloading our National Health Service at the moment may well be partly due to the fact that during lockdown, people were getting much less infection. So their immune systems are non-specifically less alert, which is what I mean.
So that load is required ongoing. We do need a certain load for the immune system to push back against throughout life.
Well, yeah, maybe. Let's treat these as two separate stories again. As far as the immunoregulation is concerned, the most important thing is getting the right organisms in during the first months and years of life, which is when you are setting up your immune and metabolic systems, and when the epigenetic changes to those systems are being organized. So early life is most important. Nevertheless, there's been a study looking at elderly people, and which has shown that as the diversity of the microbiota starts to decrease in the gut, so you start getting increased levels of inflammatory markers in their peripheral blood. So it looks as though, in terms of the regulatory pathways, yes, it's lifelong that you really want to have a good microbiota, although childhood is the most crucial period. Now, in terms of this background activation of the immune system, do you need a sort of ongoing load of infectious agents or live vaccines to achieve it? my guess is you do, but I think this whole area is relatively new, because although it was originally discovered in the 1930s, it's really only recently, with the concept of trained immunity, that people have started looking at these mechanisms with scientific and molecular detail. So that work is proceeding fast now.
And how are you arguing that we need to reassess hygiene theory?
What we've started to do is try to talk about targeted hygiene, because clearly a certain amount of common sense has to be exercised. If a child goes out in the garden and happens to have a little bit of soil on their hands when they come in, it's perfectly okay to let them sort of pick up a sandwich and get minimal amounts of that soil into their system. The organisms in that soil are pretty likely to be harmless and good for them anyway. On the other hand, if your child goes into the kitchen and spies an uncooked chicken and thinks it will be fun to pull the guts out of that chicken, that is not a good idea, because you can get nasty diarrhea and horrible infections from an uncooked chicken. So a certain amount of common sense has to be exercised. It's really a question of focusing the hygiene at the times and places where transmission of infections is most likely to occur.
And where do you think that this has gone too far? Why do you feel the necessity to actually put that in the title of your book, the reassessment of hygiene hypothesis? What is it that we've got wrong in our modern Western lifestyle that we can potentially mediate with a few changes?
Well, it's really going back to what we were saying earlier, that if you have too many cesarean sections, lack of breastfeeding, antibiotics, unchanging, monotonous diets, and lack of exposure to green space, then you are not going to meet the right microorganisms to develop your microbiota. So these are all lifestyle changes which we can deal with. For instance, if you want to take exercise, run around a field or run around a park or something where there are nice natural environment microorganisms. And of course, keeping a dog is good. We haven't mentioned that, but there's an astounding Danish study where they've looked at the likelihood of allergies occurring in children in families according to the number of cats or dogs in the house. What is slightly disturbing for your average family is that you can get to the point where the likelihood of allergies is essentially zero, but to do that, you have to have 5 cats or dogs in the house. Too many for most. But of course, you can see the logic here is that the dog, even if you don't run around in the green spaces and mess around in the soil and bring in microorganisms from the environment, the dog does. So the dog brings in the microbiota of the environment into the home. So there are lifestyle changes one can make to increase our exposure to these microorganisms. Perhaps one of the most crucial is good diet and using a minimal amount of antibiotics, as little as possible.
So let's move on to those solutions you've been talking about there. You've spoken about vaginal birth. You've spoken about breastfeeding. Presumably that is something that medical science is encouraging more and more and more. I know UNESCO are very much into promoting those sort of natural births and natural breastfeeding. Out of interest, are they using now in hospitals vaginal swabs for cesarean babies to kind of make up for the fact that they had to come out the wrong route?
Yeah, well, firstly, I should say that there's at least 20 studies showing that cesarean births sort of delay the acquisition of maternal microbiota. So there's no doubt about that, and it is associated with obesity and so on. But there have been a few studies of smearing a bit of maternal vaginal material on babies born by cesarean. I'm not sure people are terribly keen on that yet. I don't know what the current status is, how many obstetricians are doing it, or even whether they're telling everybody if they are doing it. Because I think more trials will be needed. Whereas being born by cesarean does delay your acquisition of the microbiota, does putting a little bit of maternal material on the baby's lips actually materially affect that recovery? I don't know whether we're clear about that yet. I doubt it.
And I guess this is part of the problem, isn't it? We've got to be commonsensical, but we also need to have the data to make sure that if there are changes in medical practice, we're not liable for any mistakes. I suppose that's important in medical science, isn't it? Now,
Well, absolutely. Yeah.
Another question you mentioned was the overuse of antibiotics. Now, again, this is a really fine balance that we've got to work out here. Do you feel that antibiotics are being overused? For example, in many countries, it's obligatory to give newborns a round of antibiotics. Is this something you agree with on balance? And do you think that doctors are prescribing antibiotics too much?
Well, let's start with the business of doctors. Obviously, there's a terrible temptation for a GP, an overstressed, busy GP, to prescribe antibiotics to a child or mother that come in coughing and spluttering, whereas they've probably got some virus infection which will go away all by itself. And obviously, that's not a good idea. And as I think I might have already mentioned, there's evidence that there's a linear relationship between number of doses of antibiotic in infancy and the likelihood of obesity and also of allergic disorders, for that matter. But I mean, that takes quite a lot of doses of antibiotic for these effects to get that large. So maybe it's not so important. But when it comes to the question of giving antibiotics to mothers and babies, I would be greatly against it unless there was real evidence of a significant risk of infection. And you don't give antibiotics just because you think there might be a risk of infection. And even if you give the antibiotics to the mother after a cesarean and after you've clamped the umbilical cord so the antibiotic doesn't get to the baby, the fact is that mother will now have an abnormal microbiota. So she's going to be passing a depleted microbiota on to that baby in the subsequent months. So although there have been trials suggesting that giving antibiotics to the mother, quite apart from the baby, giving antibiotics to the mother after a cesarean might be of some benefit to the mother, those trials have not looked at what are the consequences for the baby in this transaction. So again, that really ought to be done. Now, giving antibiotics once to a baby at the very beginning of its life in a country with enormously high risk of infections in that period, I think that's up to the judgment of the physician in charge, isn't it? I mean, but as I say, you don't give antibiotics unless there is a genuine high risk of infection.
Well, that was definitely what I thought. So I was very, very surprised to find that happening here in Italy, where I live. Regarding the medical training, is the data from those studies you've just mentioned about this directly proportional relationship between the quantity of doses and the problems we see later in life that you just talked about? Is that now part of doctors' training? Is that making its way into the medical schools? Because I'm just curious to find out how long this data is taking to be sort of come into the consensus.
Well, that's a very good question. As you can see from my age, my own medical training was quite a long time ago. And I have absolutely no idea what is in the medical training nowadays, except that all I can say is in a few weeks' time, I will be lecturing on exactly this topic to students in Oxford. So I guess it is creeping into the medical training somehow.
Wonderful.
Anyway, it's in all the newspapers. Every newspaper deals with this subject once a month, I would say. It just keeps on recurring.
Well, that's fantastic. I think it's long overdue that this goes completely viral. So what about solutions for my listeners who may be struggling with all of those things we've been listing, chronic inflammation, IBS, sclerosis. I mean, the list just goes on and on and on. Heart disease, diabetes, type 2 diabetes. Lots of people are suffering. Their doctors don't really know what to do apart from give them medication. Can you offer us a list of actual actions that they can take, that their GPs can actually engage them with, that could really, really help, apart from obviously the lifestyle changes that we've already discussed in detail? I'm talking about people really suffering at quite an extreme level. What can they actually do at a medical level to take advantage of this?
Well, I think, as I've rather indicated, the problem is that things like the transfer experiments that we talked about, fecal microbiota transfer into animals and so on, prove absolutely the role of the microbiota, the causative role, that it's not just symptoms, it is causative. But I've also indicated that the thing is so darn complex that we don't quite know what we're doing yet. I've pointed out that the appropriate microbiota for different individuals will be different. It'll depend on their own genetics. It'll depend on their own diet. And we don't know, as I said, whether there are certain organisms that might be quite minor components of the microbiota that might be producing molecules that are damaging to us. And we don't know whether there are certain organisms that absolutely need to be there because they're producing molecules that are necessary to us. We don't know these things yet. You have no idea the quantity of data that can be generated when you look not just at all the organisms in the gut, but actually look at all the genes in those organisms and all the metabolic pathways that this boils down to, trying to work out what is actually being made by all these organisms, or looking at all the molecules in the peripheral blood that come from the microorganisms, because a lot of the molecules wafting around in our blood, at least 30%, but nobody knows really, probably a lot more than that, are molecules made by the microorganisms in our gut. And these things are having profound effects, but we just don't know enough yet. And so I think all one can recommend to people is these rather broad behavioral things,
lifestyle.
like eating a Mediterranean diet, which seems to be, every study that compares the Mediterranean diet with other kinds of diet comes up with Mediterranean diet as the champion. And exposure to the natural environment, a varied Mediterranean diet, and exposure to other healthy people is probably good as well. But it's very difficult to be more specific yet. Of course, there are lots of companies marketing this kind of yogurt and that kind of yogurt with different microorganisms in it. A lot of that has been completely without any clinical trials to prove efficacy. But there are beginning to be glimmerings that some of these things probably are effective. But I'm afraid you're asking for more than we yet know.
And in terms of fecal transplants, then probably the most important potential treatment for the future for these extreme cases, you're telling me that there's been no human trials as yet, so we have literally no data on the efficacy.
Oh no, there have been lots of trials of fecal microbiota transplants
in humans.
In humans, oh yes, absolutely. But at the moment, relatively sort of small scale. One thing for which it definitely worked, excuse me, is people who get infected with Clostridium difficile or difficile, if you prefer, as you live in Italy, because that microorganism can be displaced by putting in a good microbiota, which gets into the ecological niche that the nasty Clostridium difficile is trying to occupy. So, that works. And fecal microbiota transplants are being tried in all sorts of other concepts. I mentioned, for instance, clinical trials happening in autism, which we are rather hopeful about, and in inflammatory bowel diseases. But you see, there's a huge problem. If we accept that the gut microbiota plays a role in the large range of inflammatory disorders that we have already mentioned, and also what we haven't mentioned in, for instance, development of bowel cancer and things like that, then how on earth do you choose your donor? I mean, okay, you could choose somebody who doesn't have type 1 diabetes and who doesn't have allergies, but you're going to have a job choosing somebody who is not already en route to getting bowel cancer in 20 years' time. So, how on earth do you choose your donor? And so, in a way, prebiotics and probiotics may be a better way to go at the moment. But again, it all requires clinical trials.
And exactly the common sense you're speaking about in terms of our personal environment. So it's really, for now, it's about lifestyle decisions, all the ones we've spoken about, and about potentially limiting our overuse of antibacterial cleaning products and of antibiotics in general. So, Dr. Rook, thank you so much for your time. This has been an incredible insight into really just bringing us up to date on where science has got to. It's so good to hear that these promising results are coming out on things as difficult as autism And I look forward very, very much to some of these becoming available on the health service. Thank you very much, Dr. Rook.
Thank you. It was great fun.